Hi friends, welcome to UNS classes. Hope you all are doing good. We are back with another previous year question. Let's see the question. Here the question is about the insulin production. The question is, with regard to insulin, choose the correct option. The option A is C-peptide is not present in a mature insulin. The insulin produced by rDNA technology has C-peptide. Option C, the pro-insulin has C-peptide. Option D, A peptide and B peptide of insulin are interconnected by disulfide bridge. It's a multi-optional question. We have to choose the correct option. So as usual, let's see what is insulin, how it is produced, what is rDNA technology, and let's come to the answer. So in your text, you might have gone through a picture like this, right? So in this picture, you can see that the pro-insulin is produced like this. In this is the A peptide. The blue color is the A peptide. The grey color is the C peptide and the green color is the B peptide. This is called pro-insulin. Pro-insulin means this is not the active form of the insulin. So it has to undergo splicing. Splicing is nothing but cutting down the protein and making it functional. So you know how protein is made from DNA and RNA is made from RNA. Ribosome will come and read the RNA and the proteins are produced. Like that, pro-insulin is produced. Now the pro-insulin has to undergo splicing to get a functional insulin. So when it undergoes splicing, the C peptide is avoided and both A peptide and the B peptide are connected with disulfide bond and it will become active insulin. Right. So in a normal body, the function is like that. The pro-insulin is produced with C peptide, which is spliced down, and A peptide and B peptide are joined with disulfide bond when we get an active insulin or a functional insulin. And when we go to rDNA technology is nothing but a gene of interest is inserted into a bacteria. When bacteria produces its own protein, the gene we have inserted will also be produced as proteins, right? So in this technology, what we'll do here, the gene of interest that we need is A subunit of the insulin gene is inserted in this bacterial DNA, and here the B subunit is inserted in the bacterial DNA. Once the DNA undergoes translation and all, it will produce its own protein plus the protein of our interest, that is B peptide here, and here it is A peptide. We will separate it out. We will take the A peptide alone from the bacterial protein, A peptide and B peptide, and we will join it with disulfide bond and we will get active insulin, right? So just see here, anywhere have we seen C peptide? No, right? So in rDNA technology, C peptide is absent, right? So if we go back to the question, you can see that the options are C peptide is not present in a mature insulin. Is it correct or wrong? Yes, it's correct, right? Second option is the insulin produced by rDNA technology has C peptide. Does it have C peptide? No, right? We have seen it, right? Just now we have seen it, only the gene of interest. We are inserting only A gene and B gene in the bacterial DNA and we are getting the peptide and joining with disulfide bond. So this option is wrong. And the third option is the pro-insulin has C peptide. Is it correct or wrong? Yes, pro-insulin has C peptide which will undergo splicing and will take A peptide and B peptide are joined to get active insulin. So this option is correct, and the option D is A peptide and B peptide of insulin are interconnected by disulfide bridges. This is also true, right? So here the option will be, see the option which has A, C and D. Here the option D is the correct answer. Hope you got the concept. Thank you. Happy learning.